A segment of DNA can be rapidly copied or amplified thousands of times with a laboratory technique called PCR, or polymerase chain reaction. The DNA of interest is mixed with a heat-resistant form of DNA polymerase, the 4 types of nucleotides, and primers. The primers are synthetic strands of nucleotides which are designed to base pair with the ends of the DNA. The mixture is heated to separate the DNA strands, and then cooled so that the primer binds to one end of the DNA. Now DNA polymerase, beginning at the primer, adds complementary nucleotides along the single-stranded DNA. The DNA sequence has now been doubled. The process is repeated as the mixture is alternately heated and cooled, and the quantity of DNA doubles again and again. Each cycle of heating, cooling, and DNA doubling takes about 5 minutes. After 20 cycles, there will be 1,048,576 copies of the DNA molecule. This technique has revolutionized molecular biology in such areas as diagnosis of disease, study of genetic disorders, and forensic medicine.
Hybridization using short single-stranded DNA molecules, known as probes, is used to identify specific DNA molecules. Strands of the DNA double helix are held together by bonds between complementary base pairs. Treatment with heat or chemicals splits the base pairs and the DNA is denatured. Probe DNAs labeled with radioactive or fluorescent nucleotides base pair with complementary regions of DNA to form hybrids.
Large charged molecules can be separated on the basis of their size using the technique of gel electrophoresis. Charged molecules will move through a thin layer of gelatinous material when an electric current is applied. Samples of material are placed at one end of the gel in individual lanes. Electrodes are attached to each end and current is applied. Molecules move toward the electrode whose charge is opposite their own. The lighter molecules move more rapidly and thus move farther while the current is on. If a dye is applied, the location of the molecules can be observed.
Bacterial cells containing specific DNA clones are identified by a technique known as colony hybridization. Cells from each colony are first transferred to a filter. Treatment of the filter with denaturing solutions lyses the cells and releases the DNA. The denatured DNA adheres to the filter. The filter is placed in a bag with labeled probe to allow hybridization to occur. Washing the filter with fresh solution removes excess probe molecules that are not specifically bound to DNA on the filter. Emissions from the labeled probe create black spots on X-ray film. Comparison of the location of the probe signal on the film with the location of colonies on the original agar plate identifies colonies containing DNA complementary to the probe.